All right, guys. Hello, everyone. This is part two of basically the lecture notes for the summer for you guys, and for chapter 13, and continuing on with what where I left off in the previous video. And if you notice in the notes, on the OneNote, I didn't put. There's a little section that I kind of skipped, or I didn't skip, but I didn't see that I missed. Right, it's essentially the crown gall disease in plants, and we talked about it in chapter 12 anyways, but I'm gonna again reiterate it again for you guys to understand what it is in a little bit more detail. Right, so we're still talking about pathogens of bacteria, right? Bacterial pathogens. So let's continue on here. So I made this little kind of annotation, 13.3.6.2, right? Before we get into fungi. Okay. All right, so let me just change a little zoom here. All right, so we're talking about crown gall disease in plants. We're focusing on a bacterium that you should be familiar with because we talked about it when we looked at modifying organisms using plants using bacteria. In this case, you have that agrobacterium tumefaciens, right? So of course, this crown gall disease is caused by this type of bacteria, agrobacterium tumefaciens. You do need to know this name, memorize this name, okay? And this is a bacteria that's quite special in the sense that it can directly pass some of its genetic material, and in the sense of the plasmid, right, into a plant, right? So it's quite interesting. It's not often that you see that in bacteria species doing that to organisms there, right? So some characteristics about this is that it could be transmitted from one host to another, right? Directly. So you can have an infected plant root, right, that has this bacteria. If the root that's infected touches another plant that is not infected, then you'll get, of course, infected there. You'll have the spread of disease that in that way. Okay, direct transmission, but it could also be indirectly, where the bacteria could be on top of certain surfaces, right? Grafting tools, car tires, soil reservoirs in the soil, right? Where you're not necessarily very close to another plant, but via humans, that they're using contaminated tools on one plant, they move on to another section of let's say of a farm or land, that they infect another plant, and so forth. Car tires, they easily can collect soil material, right, spores of the bacteria, and move it from one location to another, right? So even collecting soil from an infected area and moving it to a fresh soil that is not infected, that's the disaster happening there, right? So it could be direct or indirect transmission, and the way that you actually get infected, that portal of entry is essentially a wound on the plant, right? It doesn't necessarily even have to be a deep wound. You can have a superficial wound enough to get into that into the internal tissues of the plant, right? So most of the spread in the plants that it infects are stone fruits, right, or roses, right? Stone fruits could be like peaches, your apricots, your raspberries, right? These type of fruits, okay? And definitely this disease has been very detrimental, if I can say, for farmers and gardeners in terms of their income, and just farmers do tend to have a pretty intense financial loss due to this disease, right? So yeah, it's very important to keep in check and to monitor these diseases, right? These diseases. Okay, so in general, this bacteria can live in the soil, and or of course, the soil surrounds roots, right? And if you don't have any roots at the moment, it doesn't mean that the bacteria can't thrive, you know, necessarily having a root. It can live as an independent decomposer in the soil, or it can be in a dormant state, right? That we're going to talk about in a bit, right? For many years. And then if it has a suitable host, right, it will immediately cling on and start growing there. And one interesting thing about this bacteria is that it's very sensitive to chemical signals in the environment. It can sense, right, the bacteria can sense that a plant is injured or has gone injured, because whenever a plant does get injured, the body tends to send chemical signals to the outside, right, to the environment, and that of course diffuses out. The bacteria can pick up these signals and then grow towards that that host. Okay, so one way or another, the plant needs to get injured for an infection to occur. All right, so damaged cells release compounds that attract bacteria to the wound site. Okay. And once the bacteria gets inside, of course, it needs to reproduce. Generally, it reproduces asexually by binary fission, which you guys are familiar with. And ultimately, that constant reproduction, you start to form a what you guys already know as a gall, right? This cell mass, right, of undifferentiated cells, but this is formed because of the bacteria sending its DNA, not necessarily the bacteria itself. The bacteria infects the tissues and transmits its plasmid into the DNA of the plant cell, and it's the plant cells that begin to divide um, uncontrollably, right, to produce this tumor, this plant tumor, right, which is called a gall, a gall. All right, what is a gall? It's a cluster of undifferentiated cells, looking like a brown sphere, right? And like I mentioned before, is the result of the plasmid being integrated, the bacterial plasmid integrated into the host DNA, right? You guys already know what plasmids are. I'm not going to go into so much detail there. Okay, but of course, if you have some other form of DNA that it could alter, right, gene expression, right, or other biochemical pathways like cell division, right, maturation, differentiation, and things like that. Okay, so one way or another, you're getting your normal kind of gene expression altered. Okay, and these galls, these little kind of tumor clusters, can grow in various parts of the plant, but they generally can grow what is called the crown of the plant, which is essentially where the stem and the root join together, right? It could also grow in larger trees at the base of the branches, where it attaches to the trunk of the to the stem, right? It could also grow there. All right. So one of the biggest problems of these galls, as they grow and penetrate into the tissue of the plant, is that they disrupt, right? They interrupt normal transport of water and nutrients, basically the xylem and the phloem. And as they're doing that, of course, there's not good circulation within the plant. So the likelihood of the plant growing normally will start to deteriorate and go down and go down. So you'll likely have stunted growth, right, of the plant. Okay. Like I mentioned already, they can also form on the main stem or above the soil level or even on branches, right? So in different places in the plant. Okay. So aside from that, in severe cases where you're having the galls growing, and aside, even if you're not in the ideal environmental conditions, the likelihood of surviving incredibly goes down, that the plant most likely will die, right? So it's incredibly important to be monitoring, right, these diseases, right? So you want healthy plants, you want to avoid some type of infection there. Okay. Only bacteria that have the plasmid for crown gall can infect the plant, right? That's another thing. So you could have some bacteria that are surrounding the plant all the time, but if they don't have the plasmid, then they're not virulent. They're not really pathogenic, right? So you have to have that plasmid in order for this to occur, right? In terms of life cycle, it's essentially what I mentioned, right? If you take a look at the panel here, right? So you have these little galls, right? And the galls themselves can actually liberate spores, bacterial spores, and basically spread out. So you have these structures here, and they liberate these spores, right? And again, this is what actually passes by directly or indirectly, infecting one plant from another, right? As I mentioned before, right? In terms of direct touch, right, from one plant to another, and one plant root for another, or so forth. So as you transmit the bacteria from one plant to another, from one root to another, basically the bacteria gets attached, right? It starts multiplying. It injects or infects its plasmid into the plant cell, and then you start to get uncontrolled cell division, right? This is one individual plant, but if you see the tissues here, each little plant cell, right, that purple area is those infected cells that are starting to grow uncontrollably, and they grow more and more until you have this mass here, right, which is basically the gall. And then if you were to zoom out, kind of just to see where that that mass is forming in the root, basically something like that, and then you just have the terrible plant growing, right, inefficient. And then this gall produces new spores, which can get liberated and ultimately continues the cycle there. All right, so that's in terms of crown gall disease in plants, right? A bacterial pathogen, right? So this is the last one that I wanted to talk about in terms of pathogens. And now I want to continue on with fungi. All right, so in terms of fungi, there's two major ones that we need to know. You have chytridiomycosis, all right? And I do want to emphasize on that mycosis. Oops. There you go. Anytime you hear that word mycosis or generally you can think of it as being a fungi, right? Just to break down the words a little bit to help you remember. So in this case, you have this infection, this disease that is caused by the fungus Chytridium dendrobatides. Sorry if I'm not saying it correctly there. But in general, it belongs to this family of chytrid fungi, and they generally live in water or soil, and they mainly could reproduce asexually, right? And they are also characterized by the production of zoospores. If you guys remember zoospores, they are structures that have a single flagellum, meaning that they can propagate themselves pretty well in the water, right? Or moist soil. So, and they're produced by if you remember those little like cups or those little balls, right, with the stem, that they're the ones that liberate the zoospores. They're referred to as zoosporangium, right? So basically like a discharge structure to liberate more zoospores, right? And in particular, this disease, this fungal disease, right? Whom does it affect more? Whom is the primary host? It's essentially amphibians, right? And it has been detected at AU in Australia. It has been detected worldwide, right? It has been found worldwide. And these amphibians any amphibian that it infects, generally the population of any infected amphibian, the mortality rate is likely 100%, right? It's a high death rate. That's why it's so important to understand what it is, because again, if you're infecting more amphibians, more species, a lot of species diversity is going to decline, right? And population will decline, right? So yeah, it's at the same time, it is temperature sensitive, right? It's grown more in cooler regions, right? Southwest Australia, but still it's being shown to spread out throughout the world, right? The way that it spreads out, its mode of transmission is essentially direct via skin-to-skin contact. Okay, so there is also indirect transmission. In this case, it's more waterborne, right? Of course, you have these zoospores that can propagate themselves. So that's a little bit scary there, right? So amphibians that are in close community, that are in close range, one population with another, can greatly get infected easily, right? So yeah, you have zoospores, again, like I mentioned before, the zoosporangium, and that ability to swim and penetrate the skin and infect the skin of other amphibians, right? And if you remember from our from the respiratory system in Unit 2 that we studied, amphibians, they rely heavily on their skin to breathe, right? It's their primary respiration site, right? And not just respiration, but they also absorb water through their skin, right? So their skin is incredibly important for their survival, right? Even from immune infections, right? It's a pathogen defense system. And aside from water, if you're getting water being transported within the skin, you're also getting electrolyte being transported, right? Ions and salts and undissolved molecules there. So the skin is incredibly important to be fully functional all the time, right? But of course, the problem is that this pathogen comes in and invades the skin and invades the epidermal layers that basically comes in, damages the whole layer of tissue, right? And as it's invading and spreading and multiplying, it's disrupting, the whole respiratory system, whole salt regulatory, water regulatory systems. So ultimately, it starts to thicken, the skin starts to thicken and harden, basically even drying out. So you're basically destroying this important living system within the amphibian, and the likelihood of it surviving is greatly decreased, right? Regarding in terms of certain structures of the fungi, if you remember, fungi generally have that mycelium and that hypha, that thread-like the filaments. Not all necessarily have to have these threads, right? These hypha there. Nevertheless, they have these little filaments for anchorage and whatnot. But they moreover work as these small little spores here to easily get transported, right? And then infect new amphibians there. Okay, so their mode of replication is pretty, I don't want to say simple, but pretty quick, right? So they can infect many organisms at once, and also their virulence or their pathogenicity, right, is pretty high. So that mortality rate is pretty big. So that enables them to spread widely from one organism to another, produce more zoospores, zoosporangium, and having warmer environments and more moist environments enables them to propagate even more and more and more, right? So this does enable the fungus to spread quickly, right, and over wider distances. Okay, and of course, the hosts, in this case being amphibians, which have essentially no defense against them, right? So they're quite susceptible to this disease. Okay, so this is chytridiomycosis. All right. Oh, okay, yes. All right. So for fungi, we only have one that you need to know, right? And then we're moving on to our last one, which are malaria, which are protists. Okay, and we're going to talk about the malaria and the jarl dieback, right? So, oops, not too much, hopefully. All right. So, in terms of protists, remember these are single-celled, or they can be multi-celled organisms, right? You have this protista group, and of particular importance, not just in Australia, but throughout the world, you have malaria in more countries than others, right? And what is it, right? So it's a disease caused by a protist that is from the Plasmodium genus. So many species of Plasmodium can produce this disease, right? But of the Plasmodium species, 6 species, I don't want to say many, 6 species, right, can cause malaria. Among these, one of the most common ones, oops, what's going on here? There you go. Just wanted to italicize that. Plasmodium falciparum is the most lethal, right? And if you guys remember, when I talked about protists, there's different types of protists. You have animal-like protists, you have fungal-like protists, you have plant-like protists. So this would be a case of animal-like protists that are referred to as protozoa, right? So how does this pathogen get transmitted is essentially by the mosquito. Again, another case of mosquito transmission, right? Female mosquitoes. So they're transmitted indirectly to a host by basically blood sucking, those blood feeding of the mosquito, female mosquito. But in this case, right, you have the mosquito called Anopheles, the Anopheles genus of mosquitoes, as opposed to the Culex or the Alex one mosquitoes, right? So in this case, you have the Anopheles, very famous genus name. It's one of the more common ones that you hear, or maybe you've heard of already, Anopheles mosquito, right? And here you basically have the mosquito vector, the pathogen life cycle is jumping between the mosquito and a human host, right? So its whole life cycle requires both the mosquito, as that intermediate host, and the human host, right? That we're going to take a look at. So the female mosquito, the actual mosquito itself, is referred to as the definitive host, because here's where you would have more sexual reproduction occurring in the digestive tissue of the mosquito, right? And it enables sexual reproduction. You can have different forms of gametes, different types, the different gametes there that can enable sexual reproduction. All right. Asexual reproduction would occur in the human body between different types of tissues, from the liver at one stage and then going to the red blood cells at a later stage, ultimately then getting sucked into the mosquito again, right? So we're going to take a look at how the life cycle occurs, right? Just a little bit of information related to Australia is that it's been malaria-free since 1981, right, by the World Health Organization. So what's quite interesting is that Anopheles doesn't really occur naturally in the AU, right? So that's an interesting fact here. In 2016, right, 216 million cases have been recorded worldwide, and they've estimated that one child dies every 2 minutes from malaria cases, right? So that's quite interesting how this this disease can work, and especially on children, right? So it is a big problem that needs to be I don't want to say addressed, because they are trying to find methods of eradication, right? It's a work in progress, if I can say. All right. So we're going to take a look at the life cycle. Don't freak out from the pattern here, from the whole diagram here. Again, like I mentioned in the previous video, there's a lot more discussion in the paragraph that it's a little bit unnecessary. I think if we just follow the numbers here, it would be good enough. All right. So yeah, let's take a look. Starting from point 1. All right. And this diagram is actually it's broken down into this panel A here at the top and then panel B at the bottom. It goes into a little bit more detail between the sporozoites and the merozoites, but ultimately it's the same. It's the same thing, right? Okay, so let's begin here. So you have panel A. Sorry, you have basically number 1, where you have the female mosquito, right? And let's say this female mosquito already has the plasmodium, right? The plasmodium zygote basically means this is post-sexual reproduction, so it has a zygote already, let's say, right? And sorry, you guys, let me just, okay. In the gut, so where this is forming, right, in the gut, the zygote, remember what a zygote is basically after fertilization, where you have the female gamete and the male gamete, they fuse together, fertilization occurs to produce a zygote, right? Of course, this little single cell needs to develop, right? It's going to develop into an immature form of the plasmodium, right? Which is referred to as a sporozoite, which is an immature form of plasmodium, right? So what happens? They're maturing, they're developing in the gut of the mosquito, and then they move to the salivary gland, all right, in the mosquito. So you have the sporozoid form. They look something like this, okay? And then what happens, right? Since they're in, remember that the proboscis of the mosquito has 2 tubes. It has the salivary tube and it has the blood feeding tube, right? Since it moves into the saliva tube, right? The saliva is going to go into the host, into the human, all right? So number 2, as the mosquito comes in and bites the poor soul here, it comes in and delivers, right, the sporozoites into the bloodstream, basically via the salivary tube, right? And the sporozoites come in, and that's how we get infected. All right. Now, the sporozoites, they don't just go throughout the entire body. They do need a particular tissue in which they can continue growing, which is the liver. All right. So from the mosquito, now we're in the human, and we're in the liver, right? So within the liver, they start to reproduce asexually. So you want to have a large population growing, right? So here's where asexual reproduction is good, it's helpful, right? As they're maturing, reproducing asexually, and they're also developing more and more, right? They produce into what are called merozoites, all right? Merozoites. So you went from sporozoites to merozoites in the liver, okay? And then when they basically mature into these merozoites, they move on to another tissue type that enables them to basically mature as much as they can as full plasmodium, right? Which is the blood, right? They move into the blood from the liver, and more specifically, they move into red blood cells, okay? So here you see a bunch of these little merozoites, and they're coming in, and they get out of the liver, and they come in and infect these little red blood cells, okay? These are individual red blood cells. So, yeah, they enter red blood cells. They're still reproducing, right? Asexually, so they're reproducing more and more and more, right? You want to have many of them occurring. So to the point that there's so many inside the red blood cells that the red blood cell basically explodes, right? It bursts, it lyses, and the red blood cells, they get liberated into the blood. Each individual little parasitic cell, right, gets liberated into the bloodstream. And then what happens? You can either, these little cells either come in and infect more red blood cells, right, which happens, or they could come in and as they're developing, also one thing I want to mention is that as they're developing, they could be female or male reproductive structure cells, right? They can be male or female gametocytes, cells, right? And they're also circulating in the blood. You don't know if it's male or female, but ultimately, right, since they're flowing in the bloodstream, what happens? You get the blood being sucked in from a female mosquito, right? You have the blood sucked in from a female mosquito, and then you have these new gametophytes that are coming in. This is generally from a different mosquito. So this is why sexual reproduction can occur, right? You're not going into the same mosquito. Another little mosquito is coming in, biting you again, and it's just a whole, situation going on. And then that's why it's possible to get sexual reproduction occurring. Okay. Let me move back here. One of the things, going back to red blood cells, whenever they burst and whenever they're lysing from cells, right? Remember, they're living cells themselves, and they're doing their own metabolism, and they're liberating chemical waste from their own metabolic machinery, right? So those waste is what gives us the symptoms that we feel as sickness, right? It could be malarial headaches, chills, or burning fever, right? If you have a severe case that they're rupturing and exploding all not all of the majority of your red blood cells, that could lead to anemia, right? You can't carry oxygen efficiently, right? So it's difficult, your loss of breath, okay? You feel generally weak. to the point that it's so severe that you can get liver damage, right? An enlarged liver or spleen, and in incredibly severe cases, cerebral malaria, right, where you're having brain tissue damage, and which that can cause death. Okay, so yeah, critically important, right, to understand what malaria is and why it's important to kind of mitigate, this Plasmodium parasite. Okay, so I just want to reiterate here again, another mosquito may blood feed on the infected host, where the gametophyte can sexually reproduce in the gut of that following mosquito. Okay, so this panel B just shows a little bit more in detail of what I just talked about, essentially, right? You can take a look at that. Again, same thing. Sporocytes form in the zygote. They travel into the mosquito's salivary glands, enter the bloodstream of another host. You have these sporozoites. They come in, incubate in the liver, multiply, sexually reproduce, mature, release these merozoites. Merozoites enter red blood cells. They can continue on infecting more red blood cells and more red blood cells, or then can so what I wanted to mention, yeah, some of these sporozoids, they can mature into gametophytes, so sex-related cells, right, where they can sexually reproduce in another mosquito, and then the cycle continues and continues. All right, so a little bit more complicated, but yeah, this is the case of malaria, right? In the case of protist. Okay, let's take a look here. All right. So our last pathogen that we are going to discuss which is the Phytophthora dieback, or the Jara dieback. Okay, so this is a disease that you can see here from this pathogen. It is basically a protist that is a water mold or fungus-like protist, all right, that is called Phytophthora cinnamomi. Oh, why does it delete? Sorry. There you go. Phytophthora cinnamomi. Okay, so you need to remember that name and what it does, what are its characteristics. So this is a plant-specific pathogen, right, that affects Australia's some of major species of plants in Australia, the Jara, Banksia, if you guys remember Banksia from Unit 1. Grass trees, essentially it infects their root system, right? So in Western Australia, more than 40% of native plants are actually susceptible to the disease. So it's a very concerning disease, right? And again, I'm going to discuss the life cycle, right? And I do encourage you to take a look and read the section, read the paragraphs. If you understand the panel here with my notes, that's perfect. Remember, anything that you don't understand, please come to the classroom with questions. We can discuss, each of them more carefully, right? So, how does this work? Right, it's essentially infects the root system. All right, so again, we're talking about zoospores. All right, don't think that zoospores is just limited to fungi, right? If you notice, we're talking about protists, but they can also produce zoospores. Spores is just, you can think of it as just an asexual structure, again, that has a little filament that can propagate. Okay, so you have a plant that has the disease, right? And this disease is prevalent in the root system, right? And of course, it needs to propagate. It needs to spread out to more things, right? So in this case, you have this dieback that can produce this this zoospore. Being fungus-like, is going to have some similarities with fungi, right? So it can produce zoospore spores, and it's very filamentous, right? In the sense that it can essentially produce hypha, right? And then ultimately this whole mycelial structure, okay? And what happens here is that you have this protist that is coming in, growing in, it's infecting the root and absorbing all the nutrients from the root, right? So it's essentially killing the root cells, right? What's interesting about this case is that you don't need a wound to be you don't need a wound necessarily as the port of entry. It can attach itself from the surface. That's enough, right? But it doesn't mean that it could take advantage of any type of physical damage, right? Having a wound does enhance the whole infection situation there, right? So again, like I mentioned before, being fungus-like, you have P. cinnamomum that is thread-like, that it can produce hypha and spores. Okay, the only difference here is that, remember, for fungus, you have a cell wall that is made up of chitin, whereas this case, you have a cell wall that is made up of cellulose. So it's very similar. It has some similarities with plants. Okay, so nevertheless, you have this infection that's on the surface of roots, right? As you produce, these zoospores and they're maturing, you're having some spores that are being produced, right? These spore sacs, right, can basically the sporangia, right, can produce more spores that can infect more organisms, right? So if you take a look, they're very similar to zoosporangia, right? You can call them sporangia also. So if you zoom into these little zoospores, there's these little, I don't want to say cup, but like chamber-like structures that contain the zoospores, right? The zoospores get released, and then what happens? You infect another host plant. You infect a nearby plant, right? Now, things about these zoospores is that they can be in the soil, right? They can be thriving in the soil, and whenever it attaches to another plant, that's it. The other plant gets infected, right? The soil particle can carry these things. So you have these zoospores, they get attached to another root, and ultimately a healthy plant basically gets infected, and then the cycle begins again. All right, but let's say the conditions aren't warm, they're not humid, they're not there's not enough water, right? One way or another, it's not a good environmental condition for the spores to grow, even for the sporangia to form fully. Well, the spore can actually go a different route and become or enter in a dormant state, right? In this case, you have a state that is called climatospores. Okay, so it's a more sealed off spore, right? Very minimal requirements. There's no growth. There's basically nothing. The spore is just there and it's dormant, right? It's waiting for the conditions to be favorable so it could germinate again, so it could grow readily. So in that case, when better conditions occur, when there's more water in the area, let's say it'll start to germinate. It'll moisten. It'll get the tissue will become soft and enable it to grow, right? So these climatospores can germinate in better conditions, and then they produce the sporangia, which will produce the zoospores to infect another root, more plant roots. Okay, so depending on the conditions, either you go this route or you can go this longer route, depending on the conditions. All right. So what can enhance what is their mode of transmission and how can it be enhanced, right? Like I mentioned, it's through the root systems, right? It is moreover indirect, okay? That you have just mechanical movement within the soil, cars and vehicles in general, tractors that are picking up soil and moving it from one location to another, you're getting this form of indirect transmission, right? Just even shoes just carrying contaminated soil. If you walk on soil, right, you're spreading out. You're becoming a mode of transmission for that soil, right? And so it's moreover the disease is soil-borne, right? And it can spread more, of course, because you have moisture, you have water, the particles can move more easily, right? Whenever it rains, right? So rains does propagate the pathogen from one plant to another, right? It could be direct also, right? Root to root contact, direct root to root contact. And once you get infected, the progression of symptoms and overall maladies is pretty quick, right? One of the main things occurring is basically the root starts to rot, okay? And how that works essentially is the protist is growing, as there's an increase in protist growth and population growth. Of course, there's the plant, the protist absorbing the water and the nutrients, right, will also increase. So it will basically steal from the plant, right? Yeah, so it's basically killing the organism there. All right. So here you have Phytophthora dieback, right? It's also known as Jara dieback. Okay. Yes. So with that being said, that is basically the 10 organisms that you need to know. I know it was a lot. And I will go over this. I will make a video. So our last part 3 in terms of the factors that spread, right, the disease. And let me take a look here. But I may leave it for just this here. But I want to cover as much as I can with you guys so we can have more time to study. All right. But definitely what you need to know for the summer is the 10 diseases. All right. All right, guys. Thank you for listening. I'll be back with part 3.